Hello, my name is Candace Ward. In this video, we'll start building your knowledge of enzymes. We'll only brush the surface of basic enzyme function here, but we'll make sure you understand enzyme structure and classification. To start off, we'll begin building a protein from its most basic layer and working out to increasing complexity. We'll explore how enzymes function to catalyze biological processes and then break down the 6 classes of enzymes: oxidoreductases, transferases, hydrolases, lyases, isomerases, and ligases. We'll familiarize you with both the general function and nomenclature of each class of enzyme. On the right side of your screen, you'll see a molecule key. We'll follow this key throughout the video, and I'll remind you of any atoms or bonds of note. The main aim of this video is to familiarize you with the different types of enzymes and their corresponding characteristics. That way, you should be able to see the name of an enzyme, like hexokinase, and deduce certain characteristics about the enzyme itself. So by the end of this video, you should be able to answer questions like the one at the bottom of your screen that ask about common characteristics of certain groups of enzymes. Let's lay down the basics. What is an enzyme? An enzyme is a protein that functions as a catalyst, specifically in biological processes. Without enzymes, our bodies would have a hard time completing normal chemical processes in a useful time frame. Enzymes speed up these processes, as we'll examine later. A key characteristic of enzymes is that they remain unchanged after a reaction. So an enzyme should appear both in the initial and final stages of the reaction in the exact same state. In addition to the traditional definition of enzymes, there's an offshoot known as ribozymes, which is a bunch of RNA that acts as a catalyst. The categorization of ribozymes as enzymes is somewhat controversial, but it's important to recognize that there is an exception to the hard and fast definition of enzymes as proteins, since the MCAT could throw information about a ribozyme at you in a passage. Now we know that both enzymes and ribozymes work as catalysts to speed chemical reactions up. However, these catalysts can't go jumping around the body speeding any old reaction up. A noted characteristic of enzymes is their specificity. This means that one enzyme can only affect a small number of molecules. The molecules that are affected by an enzyme are called substrates. In our ribozymal subunit we have on the screen, we see exactly where our mRNA would fit into the active site to undergo translation. The active site is where all the action happens. This is where the enzyme can change the molecule to affect the chemical reaction. Now that we've covered basic enzyme characteristics, let's take a look at the underlying structure that allows enzymes to function. Since our main focus in this video is enzymatic action, this will be just a quick review of protein structure. Primary structure describes a chain of amino acids, linked by peptide bonds. We can see a peptide bond forming in our figure here at the bottom. Many peptide bonds together create a polypeptide chain, which is our primary structure. Secondary structure is the configuration of these polypeptide chains into cool structures like alpha helices and beta sheets. Next, we have tertiary structure, which describes how these secondary structures are arranged in relation to one another. And finally, we have quaternary structure, which details the arrangement of multiple subunits in a protein, like the dimer seen below. Now that we've figured out how to describe the structure of an enzyme, let's get down to the nitty-gritty of how they function as catalysts. Catalysts work by lowering a reaction's activation energy. We can think of chemical reactions like rolling a ball down a hill. In order to roll a ball down a hill, we have to climb the hill first. Climbing the hill is our activation energy. It's the amount of energy we need to put into the system before we can get our reaction. And in our nifty graph at the bottom right, that's what we'll see. If we start out on the left, we have to climb that hill in order to continue down onto the right. As you may remember from general chemistry, the change in energy across a reaction is called Gibbs free energy, represented by the delta G. Here we're going from a situation of higher energy. We're going to climb the hill to reach our activation energy and then reach our product of much lower energy than our initial reactants. The way our enzymes work is to better prepare our substrates for this change. If our total reaction looks like this, with an initial state and then a more stable final state, it makes sense that it has to go through some configuration that's somewhere in between the two states. This middle configuration is called a transition state. You should notice that this transition state is at the highest energy and is therefore the most unstable. Our molecules don't like to be unstable, so it takes more energy to force the molecules to adopt this configuration. However, when our enzymes force the reactants into the transition state, the reaction proceeds faster, since the hard part is already done. A fact that's essential to know for test day is that enzymes function only to affect the kinetics of a reaction. They can only make a reaction go faster. They do this by lowering the activation energy. To go back to our metaphor, we now have a smaller hill to climb in order to roll our ball down the other side. It's important to distinguish between the thermodynamics of a reaction and the kinetics. Enzymes only affect how quickly a reaction reaches equilibrium. They don't change the enthalpy of the reaction. Something else to note is that these chemical reactions are reversible. In this case, the reverse reaction has a much higher activation energy than the forward reaction, which means that the reverse reaction occurs more slowly than the forward reaction. However, when our enzyme catalyzes a reaction, the activation energy of the overall reaction is lowered, including the activation energy for the reverse reaction. So while we're getting from left to right faster, we're also increasing the likelihood that our reverse reaction will occur as well. Enzymes do not affect the ratio of products to reactants at equilibrium, since our reverse reaction is affected just as much as our forward reaction. This means that we get to that equilibrium faster. We reach that balance of forward and reverse reaction rates quicker than if we didn't have a catalyst. Now we should have a better understanding of how all enzymes work. They work to lower the activation energy of a reaction, decreasing the amount of energy that needs to go into a system in order for the reaction to take place. But, of course, not all enzymes catalyze the same reaction. Enzymes are categorized according to the general type of reaction they catalyze, and we need to know 6 different types of enzymes for test day. The first 2 types of enzymes we'll cover are oxidoreductases and transferases, which both shuttle things around. Next, we'll explore hydrolases and lyases, which both cleave molecules. And finally, we'll deal with isomerases and ligases, which affect the bonding of molecules. We'll use a convenient color scheme to help differentiate between the enzyme types on the slides to come. If you're a bit intimidated by all these enzyme classes, don't worry. Calvin has a pretty great mnemonic to help you remember the 6 classes. Lil hot. Ligases, isomerases, lyases, hydrolases, oxidoreductases, and transferases. Keep this mnemonic in mind when you study so you can integrate these enzyme types into your memory. To start off with, we'll examine oxidoreductases. Just by looking at the name, you could guess that this enzyme class has something to do with oxidation and reduction. Now, oxidation and reduction are 2 reactions that are the opposite of each other. Oxidoreductases catalyze the transfer of electrons between biological molecules that contain elements like carbon, hydrogen, and oxygen. In our molecular diagrams in the chart, black represents carbon, red is oxygen, and white is hydrogen. Moving electrons around can make atoms more oxidized or reduced, as we see in our chart on the screen. Reducing a molecule means that we have fewer bonds to oxygen and more bonds to hydrogen. Here we see the reaction another way. Adding bonds to a hydrogen is a reduction, and adding bonds to an oxygen is oxidation. Oxidoreductases catalyze oxidation and reduction, and they typically have help from molecules called cofactors. Electrons can jump on and off molecules like NAD+ and heme. In the figure at the bottom left, we see NAD gaining and losing an electron in the form of a hydride ion. In this case, it's helping an enzyme called GAPDH during a redox reaction by carrying an electron. Heme has the same function in an enzyme called cytochrome C, which you may be familiar with if you really love the electron transport chain. Heme is located at the center of this enzyme and helps with redox reactions during aerobic metabolism. Some examples of oxidoreductases include those listed on the screen, and they can sometimes be tricky to identify. Oxidoreductases are ubiquitous, so if you have no idea of the function of an enzyme, this enzyme class is a good guess. In contrast to oxidoreductases, transferases are much more obvious in their function. Transferases shift functional groups between independent molecules, and they often use coenzyme donors. In our reaction schematic at the top right, we see functional group B being taken from A and added to C. A transferase could catalyze this reaction. A transferase could work like this. Here we have an alanine loaded into the active site of an alanine transaminase. We have this amine group on the left side of the alanine being transferred to the left side of the alpha-ketoglutarate, and an oxygen from the alpha-ketoglutarate takes the place of the amine group on the alanine. As a result, we get glutamate and pyruvate. Unlike oxidoreductases, the nomenclature for transferases is a bit more straightforward. You'll usually see transferases named in one of the 2 ways listed on the screen, with the words functional group being replaced with something like amine or acetyl to describe the functional group being moved. Here are a couple of examples. A noted exception to this rule that you'll need to know for test day is kinase. Kinases are transferases as well, and catalyze the movement of a phosphate group. ATP typically helps kinases as a coenzyme donor, giving up a phosphate group as indicated by the arrow. Another exception to a typical transferase nomenclature is polymerases, such as DNA polymerase 1 or 3. Polymerases shift nucleotides onto growing strands of DNA or RNA in the case of RNA polymerase. While transferases may change what the substrates look like, both initial compounds are technically still present after the reaction. In contrast, enzymes that cleave molecules may eliminate some products. One type of cleaving enzyme is hydrolases. Hydrolases add water into a reaction, causing a molecule to break into its components and bond to the hydrogen molecule and hydroxide group, as we see in our reaction schematic in the top right. Pepsin is a hydrolase. Here, we see pepsin bringing a water molecule into contact with a polypeptide chain, where it will break a peptide bond, creating 2 separate chains of amino acids. Now, water obviously doesn't cause all molecules to break apart. However, macromolecules that were synthesized by dehydration synthesis will be broken apart by hydrolases. Sometimes you'll be lucky enough to get a hydrolase with the word hydrolase in its name. Unfortunately, we can't always be that lucky. Sometimes hydrolases will be named simply for the substrate it affects, like peptidases, which catalyze hydrolysis of peptide chains, nucleases, which catalyze the hydrolysis of nucleic acids, and so on. Many digestive enzymes are hydrolases. We've already seen pepsin, and trypsin is another digestive hydrolase. On the MCAT, you may be asked to describe the reverse reaction of a process you've been given. We've just seen that hydrolases use water to split molecules apart, and the reverse process, where water is removed from a molecule, is known as dehydration synthesis. We just covered enzymes that split molecules with the help of water. Lyases are enzymes that split molecules without water. When these portions are broken off, they tend to form rings or multiple bonds in order to re-establish the octet of electrons around each atom. Let's look at an example. Here we have adenylate cyclase catalyzing the lysis of ATP. Two of those phosphate groups break off, yielding inorganic phosphate, but the final phosphate group remains attached to the adenosine. We actually get a cyclic structure called cyclic adenosine monophosphate, more commonly known as cyclic AMP. And you probably could have predicted that we get a ring in our product purely from looking at the name of the enzyme. So we could have our lyases simply named with the substrate and lyase coming after it. However, think back to how enzymes affect both the forward and reverse reactions. Lyases can function not only to lyse things apart, but they can put molecules together too. Lyases may also be named for the product they create, with the word synthase following it. Lyases differ from other enzymes in this regard. They require only one substrate when they're cleaving a molecule, but they require two substrates for the reverse reaction. Let's look at another lyase reaction. We'll start off with fructose 1,6-bisphosphate and catalyze its cleavage with aldolase to form DHAP and glyceraldehyde 3-phosphate, more commonly known as GAP. On test day, you may encounter enzymes that don't cleave or merge molecules. Isomerases, for example, rearrange the atomic connections within a molecule. We still have DHAP and GAP on the screen, and you should notice that they both have the same chemical formula, C3H6O6. However, they have different connectivities. Atoms are connected differently in each molecule. An isomerase can catalyze one molecule to reconfigure itself into another molecule entirely. When we switch around the atomic arrangement of a molecule, we're flipping between constitutional isomers and stereoisomers. You may recognize these terms from organic chemistry, and we go more in depth on this topic in your organic chemistry resources. Back to our two molecules, DHAP and GAP. TPI stands for triose phosphate isomerase, and its substrate is DHAP. GAP is one of the molecules in glycolysis, and the enzyme that converts GAP to the next step only recognizes GAP, so we somehow need to change DHAP, which has the same chemical makeup as GAP, into GAP itself. TPI flips some atoms around, and now we have two molecules of GAP to be used by GAPDH further down the line. So isomerases are labeled either as isomerase or as a generic substrate with the enzyme ending of ACE. Isomerases frequently involve shifting electrons or cleaving bonds, so they may actually be considered oxidoreductases, lyases, or transferases as well. Almost done! Our last class of enzymes is ligases. Ligases use ATP to link biological molecules. We generally see ligases functioning during DNA synthesis or DNA repair. Since you probably already know the process for DNA synthesis, it'll come as no surprise that ligase is used to create bonds between Okazaki fragments. Once DNA polymerase is done synthesizing these fragments, DNA ligase descends upon the lagging strand. Using the energy from ATP, the fragments are fused together to create a whole daughter strand. It's important to note here that while other enzyme classes can hydrolyze ATP, ligases are the only class of enzyme that absolutely needs ATP to function. Now, on to the nomenclature. Ligases can be called something synthase or something synthetase. Synthase can be a tricky thing in nomenclature. It simply refers to something being fused together. And earlier we realized that lyases catalyze the reverse reaction, thereby taking two molecules and fusing them. Those lyases could be called synthase as well. However, synthetase is a name that is specifically used for ligases. This difference isn't essential to know for test day, but it could certainly save you a bit of time if you know it. It took a lot of hard work to get to the end of our content for this video. How about we test our newfound knowledge with some practice questions? Here we're asked about the similarities between enzymes in the same class. Take a second and pick an answer. The answer is B. Enzymes in the same class typically perform the same function. All ligases form bonds, all oxidoreductases catalyze redox reactions, and so on. How about another one? Here we need to select one or more statements that accurately describe enzymatic function. Take a second and pick an answer. The correct answer is A. Enzymes function by reducing the activation energy of a chemical reaction. Roman numeral 1 is false because high temperatures typically denature proteins, rendering our enzymes useless. 3 and 4 are false because we noted that enzymes do not affect the thermodynamics of a reaction, only the kinetics. Practice question 3 asks us to identify the simplest level of protein structure. Take a second and pick an answer. Primary structure is the simplest level of protein structure, since it's just a straight chain of amino acids. Keep up the hard work! We're asked to identify what kind of enzyme isomerases cannot be co-classified with. Take a second and pick an answer. If you recall from earlier, isomerases involve bond breaking and electron shifting, so they may also be considered hydrolases, lyases, or oxidoreductases, but not ligases. Alright, let's finish strong with one final practice question. We need to classify ATP synthase and explain why it fits in that category. Take a second and pick an answer. The correct answer is D. Earlier we realized that synthase could refer to either a ligase or a lyase. ATP is synthesized by combining ADP and inorganic phosphate, two dissimilar molecules. Ligases typically create bonds between two of the same molecules and require ATP, so lyase fits the question better. We covered quite a lot in this video, so let's take a minute and lay out the main takeaways so that we know what we learned. Enzymes are pretty amazing molecules that aid in a huge variety of biological processes. We explored how enzymes decrease the activation energy of a reaction, therefore increasing the forward and reverse reaction rates. The equilibrium constant, which is the ratio of products to reactants at equilibrium, remains unchanged with the addition of an enzyme. And we noted earlier that enzymes do not affect the thermodynamics of our reaction, so the Gibbs free energy remains the same. We covered the 6 classes of enzymes. Oxidoreductases catalyze redox reactions. Transferases shift functional groups from one molecule to another. Hydrolases use water to both cleave and create bonds in hydrolysis or dehydration reactions. Lyases cleave and create bonds without the use of water. Isomerases change how atoms are connected within a molecule. And ligases create bonds between large molecules and require ATP to function. Awesome job keeping up with all those enzymes. We explored some really cool molecules in this video, but don't worry if they haven't stuck in your mind yet. Keep practicing, and thanks for watching.